So good afternoon, everybody, and welcome to today's vegetable industry webinar, where we'll be looking at priority pests and diseases in the Werribee region. My name's Maddy Quirk from AUSVEG, and I'll be facilitating today's session. Before we do get into the program, I'd like to begin by acknowledging the traditional custodians of country throughout Australia, as well as their connections to land, sea, and community. I'm on Wurundjeri country today, but wherever you're joining us from, may we acknowledge country from which we produce our food and its people who have looked after and survived on this land for thousands of years. We pay our respects to their elders past, present and emerging, and extend that respect to all Aboriginal and Torres Strait Islander people here today. This webinar is brought to you by the AUSVEG Peri Urban Biosecurity Pilot Program in partnership with Agriculture Victoria and NielsenIQ. The program is funded through the Department of Agriculture, Water and the Environment. Through this project, we're focusing our efforts to support your business through strengthening pest and disease management, encouraging testing of exotic pests and diseases and pest and diseases of concern, and providing support on how to do this so that as an industry, we can try to control or contain pests of concern and minimise their long-term impacts on growers. So first of all today, I'd like to announce to you an initiative that AUSVEG and Agriculture Victoria have been working on together called the Anonymous Reporting Pilot, which aims to enhance vegetable biosecurity through anonymous reporting of suspect pests. The pilot project provides a secure and anonymous process for growers and agronomists to confidentially submit samples of potential exotic pests and diseases to Agriculture Victoria's Crop Health Services for diagnosis, which is currently the process that's in place at the moment as well. If you choose to send a sample through this anonymous reporting system, your report will remain anonymous through the entire diagnostic process. And if the sample returns an exotic result, meaning that it was diagnosed as a pest or disease that is not currently in your production region, only then the result and identity will be reported to the Victoria's Chief Plant Health Officer, and direct communication with the growing operation will then commence. If an exotic is excluded, this will be reported by the Crop Health Services directly back to you as normal, and you will be given the option to further diagnostics if you require advice to further inform pest and disease management. So how has this changed from the current process? Well, the benefit of this pilot is that a business will not be negatively impacted during an investigation phase to determine if the report was an exotic pest. The business will be able to continue without regulatory action while the diagnostics are being undertaken. The anonymous reporting sample submission form is in final draft stages, but it will be available for use very soon. And it is a tailored form that is targeted at capturing relevant data to Crop Health Services and has been enhanced to do this. For more information, you're welcome to contact myself or Zarmeen Hassan, and our contact details are on the screen here. The form will be accessible via the AUSVEG website and promoted in upcoming AUSVEG newsletters in coming weeks. So today we have a versatile array of presentations, including an announcement about the Vegetable Extension Network program from Danielle Park AUSVEG, key consumer trends to look out for as we transition into the next phase of COVID by Llew from NielsenIQ, a Silverleaf whitefly and tomato yellow leaf curl virus situation and market access update by Lavinia Zirnsak from Agriculture Victoria, an update on vegetable virus diseases in Victoria by Kinoti from Agriculture Victoria, fungal and bacterial diseases across the Werribee region by Ramez Aldaoud from Agriculture Victoria, a video on exotic leaf miners that I developed, and our consultant John Fletcher assisting with Q&A.
Thank you, Maddy. So just to provide a bit of a brief update of a few changes that have occurred with the VegNET program at both the national and also the Victorian level. So this is some information from October 2021. So, and there's a new regional development officer, that's myself. So my name's Danielle Park and I work with AUSVEG. I've worked as the EnviroVeg program coordinator for the last 2.5 years with AUSVEG. And I've worked very closely with Dimi Kirikou through that time, forming connections with growers in the region. Back on Friday the 1st of October, there was a change to the National Extension Program, the VegNET program, where it's moved to being managed by AUSVEG. AUSVEG is the peak industry body for vegetable growers and will lead VegNet for the next 5 years, liaising with existing RDOs to ensure a nationally consistent, regionally tailored, industry-focused extension program for the benefit of vegetable growers. RMCG will continue to contribute to VegNet 3.0, which is the current project. RMCG has successfully delivered VegNet in the northern, western and southeastern regions of Victoria over the last 6 years. It's helped growers improve their growing practices and their profitability. RMCG is going to work with AUSVEG as an independent monitoring and evaluation manager for the national VegNet program, and they will also continue to deliver VegNet in Tasmania. With RMCG's change in role, or their expanded role within the program, it's been agreed mutually that the transition, the RDO role to AUSVEG from RMCG, and this will ensure that future extension for the regions will build on the strong foundation that have been set by DIMI and RMCG, and growers will benefit from AUSVEG's industry networks and close ties with local growers. DIMI would like to pass on a farewell and a thank you at this time. So she's very keen. She will continue working with RMCG and work to the benefit of horticultural sector, including vegetables, through the Soil Wealth Integrated Crop Protection Project. She wanted to pass on that she's enjoyed delivering VegNet in Victoria in 2021 and looks forward to continuing to engage with growers and industry to help growers benefit from levy R&D investment and improve their farming practices. At the moment, we're in a transition phase where we're moving some of the work that's being delivered by RMCG to AUSVEG. We'll work collaboratively to ensure there's a seamless transition and that we continue to support Victoria's vegetable growers and connect them to levy-funded R&D. We're looking to foster innovation and best practice on farm. That remains our key feature. And from a grower's perspective, it will be very much business as usual for VegNet in Victoria. So the main change is that the point of contact will be a little different, and that will be myself. I'm looking forward to learning more about the Victorian vegetable growing industries. My role in the past has been national, which has meant that I'm often travelling from place to place. It'll be great to be able to build a deeper knowledge of the systems and opportunities for the vegetable industry in Victoria. Thank you.
All right, thanks everybody. Thanks for having me here. It's my first time coming to one of these. So I'm Llew Stevens. I'm the new Fresh Industry Lead for NielsenIQ in Pacific and Australia. I've been with Nielsen about 10 years and pretty much all that time I've been working on Fresh, mostly for the retailers before this. So what I want to take you through today is a bit of an overview of sort of what we've seen over the last couple of years, particularly in Victoria, and some of the key changes we've seen in the market that will have implications as we move forward and we start reopening and we move into post-COVID, quote unquote, normal. We're never going to get back to pre-COVID normal, so it's really about where things are going. So a little bit of a note as to what NielsenIQ home scan is, which is the data source I'm using today. So this is a panel of households that we manage internally that's representative of the Australian population. We get our panelists to scan everything they bring home for in-home consumption, and then we aggregate that up and use it to represent the behaviours of the overall Australian population. This data is used heavily by manufacturers, retailers all across Australia and globally. It's pretty much the same way we would do something like TV ratings that we do it for food. So as a quick overview, obviously there's a lot of text on this slide. Let's give you a moment to breathe it in. So there are three key trends as we move through COVID and we move past COVID. These are kind of trips. That's kind of the amount of times people go to a supermarket or a store in general, online, and then basket value. So these are some of the three big shifts we've seen over the last couple of years. When I go through this, generally I'm talking about the 52 weeks to September versus 2 years ago, because I don't want to talk too much about the COVID year because purchasing was so extreme in that year. So it's about where are we versus where we were before. So at an Australian level, we can see that trips per household have declined versus the pre-pandemic levels. Doesn't seem like a lot, only 2.2 fewer, but that does add up across the overall population. As we go down, we're looking at Victoria. It's actually even more pronounced in Victoria. And given the amount of extended lockdowns we had in Victoria as a fellow Victorian, it's not surprising that we've pulled back on the number of trips we're making out and about. We're down 5.4 trips, still shopping quite a lot though, more than the Australian average, which is quite interesting. Moving across, in 2019, before the pandemic even hit, I remember talking to lots of clients about how much online was growing, how fast it was growing, how it was the outlet of the future. But really that's accelerated quite fast throughout the pandemic to the point where it's basically doubled in size in terms of its share of overall FMCG in the 2 years since. In Victoria, that's a bit faster again, up 5.1 percentage points, still at almost 10% share of overall FMCG. And the final one is basket value. So as we see reductions in trips, typically we would expect an increase in basket value. Because obviously people are shopping around less, they're probably buying more when they do shop. What we're seeing is quite a large increase in the size of the baskets overall, up over $5 for the average Australian basket of goods. In Victoria, that's bigger still, up $6 to an even larger one. Part of this as well is prices are increasing across a variety of commodities. And I know particularly in the fresh space, this is probably something that is top of mind for a lot of producers, distributors, and even consumers. So this is something that we're going to have to face into as we move into the next phase of COVID. Taking the next step forward, what I've got here is a trended 4-week view of average trips per household. In the green, I have Victoria, the number of trips made in an average 4-week period for households in that 4-week period. In the black, the total Australia average. And then in grey, I've got New South Wales. That's a pretty nice comparator to Victoria, given our proximity and given what we've been going through over the last 6 months. On the left-hand side around March is that big spike. What we saw in the initial Australian lockdown was a huge spike in the average occasions per household, where people were forced to shop around for their goods due to the product limits and things like that. As we moved into the Victorian lockdown, we can see Victoria, which was higher than the Australian average, came down kind of in line with the Australian average, but still kept shopping at or slightly above the overall Australian average. But then as we move it over on the far-hand right-hand side, we can see New South Wales's most recent lockdown, where you can see them go in a rather large dip, similar to what Victoria went through. So I think one of the interesting things here is that Victorians just shop more in general than the Australian population. And that's something that's kind of seen us through even the heavier restrictions. And that's something we probably could expect to see continue going forward, but at probably a lower rate than we saw before the pandemic. Taking the step then into vegetables. What I have on the left-hand side for you here is the share that online makes up of total vegetable sales in the green, and in the grey, the share that online makes up for total FMCG. So you can see when I was talking before, it's hit about 10% on the 52 weeks. On the 4 weeks, it's even more pronounced, up to almost 12% of share for total FMCG. And we can see online vegetables is generally following that trend. So its adoption for fresh online has always been a bit more difficult to get customers across. People tend to like touching things, picking things up and assessing their produce themselves, but it's still following the overall trend. So as online picks up, we would expect produce to continue seeing more and more sales come through online. On the right-hand side, we have the value percent growth versus a year ago for both total online vegetables and total online. So you can see the high growth throughout late 2020 on the left-hand side there. And that's dipping off as we go into the same period this year, as we try to cycle that growth. Still growing 33% on top of 106% the year before is still extremely high growth and one of the key drivers of overall market expansion. To look at that then in Victoria versus New South Wales to see how we're going. So this is again just value percent growth for online, for total online vegetables, looking at total Australia in the black, total New South Wales in the grey, and then total Victoria in the green. So you can see in our lockdown, we saw an extremely high growth in online. We were utilising a lot more than we had the prior year. And then as we came out of that lockdown, we settled more in line with the overall Australian average for growth. And then as we moved into later this year and started to cycle that growth, we're actually quite flat in terms of our adoption of online. But at the same time, New South Wales was entering on lockdown and seeing very similar trends to what we saw in our lockdown too. So again, we can expect to see online adoption to continue at quite a high rate. In marketing, we always say trial precedes liking, and after 2 years of people engaging with online, we can probably expect some behaviors to stick around as we move forward. Moving on to kind of see how Victoria in particular compares with other markets. So what I've got for you here is a driver of growth for the 52 weeks versus pre-pandemic. And so how do I do drivers of growth? Well, I take the overall growth in the market in dollars, and I understand what contributed to that growth. Was it frequency? Was it shoppers shopping more often? Was it volume per occasion, picking up more produce every time they go in? Was it the price per volume change? Or was it the number of households? And so that's, more people coming in, population expansion, things like that. And what we can see across the vast majority of the market is this increase in kilogram per occasion and price per kilogram. That's quite consistent across most of the market. With basket, and these contribute together to become that basket size conversation I was talking to you about before. Victoria kind of stands out as one of the few states where kilogram per trip is outpacing price per kilogram growth. Price per kilogram growth, you can see in the grey on the left-hand side of total Australia there is probably the single most significant contributor to overall growth. I was also asked to kind of talk a little bit about some of the specific varieties of interest, notably broccoli, lettuce, celery, leek, carrots, and pumpkin. So this obviously is a lot of numbers, so I'll leave this up as I kind of walk through it a little bit. So on the left-hand side, we have the growth versus 2019, both in kilograms and dollars. This is just for Victoria, mind you. Then we have the percentage of Victorian households and how that has changed over the last couple of years. The average trips per household, the spend per kilogram, and then the kilogram per trip, which is essentially the KPIs we went through in the previous slide there. Overall, what we can see is that across all these varieties, there's quite good growth versus 2019. There probably were some peaks and troughs over the last 2 years, though. The key thing that I noticed going through this is good growth in average trips per household, but some slight softness maybe in the percentage of penetration of the household engaging with it, while kilogram per trip stayed pretty consistent, even with some spend per kilogram increases. I think this is where one of the key considerations, which I've talked about with other producers and other organizations, as well as with these changes in how people are shopping, how they're shopping around, things like longevity on shelf, freshness, and things like that become much more important, potentially become much more important to consumers as they are engaging with the category a bit less. They're less able to go in and do that quick top-up shop for fresh if they're focusing their shop on, let's say, one major retailer or something like that. So that's thank you. That's it for me. My contact details are on screen, and feel free to reach out if you have any queries.
So thanks, Maddy. I'm Lavinia Zirnsak. I'm a Senior Policy Officer with Domestic Quarantine, so working in the market access area. So Maddy asked if I could give a bit of a rundown on the current situation of, I just realised I didn't have my video on, the current situation of tomato yellow leaf curl and silverleaf whitefly. And it's good that I think I've got Cliff following on to me, so he might actually cover some more of the sort of management and biology component. So the background is basically we had partial state freedom for silverleaf whitefly in Victoria. It was a 50k restricted zone up around the Murchison Shepparton area up in the northeast, and basically we had that radius there for, I think it was probably a good 10 years in terms of a restricted zone where things that moved out of that zone had to move in accordance with entry conditions from other states, because we knew that silverleaf whitefly was there. We were probably quite lucky to maintain silverleaf whitefly freedom for as long as we did, considering that we probably, by the nature of silverleaf whitefly and its movement patterns, it probably moves a bit quicker than we kept up with surveillance there. So COVID presents difficulties and opportunities in most climates. We were presented with an opportunity from Western Australia this year to do some remote surveillance via a business to be able to move product, because under the COVID conditions, we couldn't actually enter their premises. So we were able to work with them to do their own surveillance, but what that meant was more robust diagnostics. And through that more robust diagnostics process, we actually picked up quite a few samples of silverleaf whitefly down in Gippsland and a few others. So ultimately, we could no longer defend the position of area freedom. So as of the 1st of August, we actually lost our area freedom for silverleaf whitefly, which means that it was then moved to the established pest list within Victoria under the Plant Biosecurity Act. So we couldn't meet national biosecurity requirements to keep that up and going. We informed the other states, and then as market access, we go into discussions with other states to try to work out entry pathways into those states. So basically, producers consigning material to Western Australia or Tasmania must ensure that they're treating their product in accordance with the conditions that are set by those markets. Basically, there is anyone who's producing vegetables and sending some of those sensitive markets, they're probably familiar with ICA 29, PS 27. So there are arrangements that we've been able to negotiate with other states for entry. WA has an entry requirement of presenting produce on arrival, and they do another inspection to sort of add confidence for product coming into those states. So the good news is that silverleaf whitefly is already established in New South Wales, Northern Territory and Queensland. So there's no conditions moving in there. And clearly, if you're looking at, on the flip side, as a vegetable producer, getting seedlings from those areas, they're sort of free to move down. It's obviously good practice to try to be looking for high health seedlings when you are planting or planting from seed. Because ultimately, you know, you just want to make sure that your seedlings are hopefully not bringing silverleaf whitefly down, because they can also be carrying tomato yellow leaf curl virus as well as the vector for that virus. So Tasmania and South Australia currently have, they claim state freedom for this silverleaf whitefly. And pretty much Tasmania and WA have never really accepted our partial area freedom status anyway. So when we did an analysis with industry, we actually talked to AUSVEG, NGIV and other industry bodies to see what the impact of removing silverleaf whitefly would be on industry. And it was deemed that due to protocols being in place, there probably wouldn't be too many market disruptions. Okay, so these are the families. So your Forbiaceae, I'm going to mess this up, but obviously the spurges, more of the nursery type plants. The main one in this area is kind of poinsettias, and apparently Victoria sends, I think, the majority of poinsettias to Tasmania. So that means no poinsettias in Tasmania for Christmas unless Tasmania help us with a protocol to get them across. Obviously convolvulaceae, which has, you know, sweet potatoes in there, cucurbits, obviously, your pumpkins, squashes, beans in your legume family, your solanaceae, obviously your tomatoes, and this one's mainly nursery plants, I believe. So these are the plants that tend to be affected when you're moving interstate for silverleaf whitefly. But as indicated, there are protocols that can help you get there. So the second part of the presentation is, so basically, in a nutshell, we've lost freedom for silverleaf whitefly, but there are protocols that can help you with interstate movement. So tomato yellow leaf curl is the virus which silverleaf whitefly is the known vector for. You'll probably hear more about that from Cliff's chat. So basically in April this year, we actually had a notification from Queensland Ag Department to let us know that they'd had samples from Victoria that were submitted to Queensland, and they had a positive sample from tomato yellow leaf curl. So whilst tomato yellow leaf curl is not exotic to Australia, it was exotic to Victoria, so it does fall under our exotic legislation. So basically through some surveillance, we've found out that there is 3 positive areas of production in northern Victoria there. The seedlings have come from 3 separate facilities, some from Victoria, some from WA and some from Queensland. The ones that came from Queensland came down on permit, so they'd met their biosecurity obligations, and WA apparently doesn't have it. So, and we're also doing further tracing. So we basically had to let our sensitive jurisdictions know that we found tomato yellow leaf curl. Tasmania has a 20k restricted zone condition in there, so they want to know that any facility is 20 k's from a known detection. And WA had a 500-kilometre zone, which would have knocked out most of the state. They've been very collegiate and they've actually reduced it to 20 kilometres where we undergo a delimiting surveillance program to sort of see how far the spread is in Victoria. So this is just, sorry, being PowerPoint, it's probably not big enough, but sorry, this is the map we have. So it's this area up around here, which encompasses Numurkah, Kattunga, Cobram, Nathalia, up around there. So in that northeast part of the state. So that's currently got a 20-kilometre restricted zone on there that is being gazetted. So any movement of product outside of this zone requires certification and treatments to manage the risk. So we developed a surveillance strategy to try to gain situational awareness of tomato yellow leaf curl virus within the state. So we divvied that up into phase 1A and 1B and phase 2. So phase 1A was looking at high risk facilities, which were obviously hothouse tomato production because they're available at the moment. They provide a good habitat for silverleaf whitefly if it exists in them. Also nurseries, they were deemed high risk because they're sending outside of Victoria, so therefore it's a risk for particularly WA but other states. And then part 1B was looking at other hydroponic production, and phase 2 is around our field grown tomatoes. So obviously our processing tomatoes up north, and any other sort of field growns that we can pick up through our investigations. So far, the results have been promising in that, sorry, this map's a little out of date because we get information all the time, but you can see here the red is the positive detection and all the other surveillance points that have been done in 1B so far have been negative for the leaf samples for which Crop Health Services have been doing for us. And ultimately, I think at the moment we're up to about 14 or 15 negatives. So we haven't had a positive detection outside of the 20k restricted zone. Western Australia have amended their entry conditions. So basically anyone importing nursery stock of those families into WA need to adhere to condition 26. In saying that, Truss tomatoes are actually exempt because they redid a new pest risk analysis and they were deemed below ALOP. So the risk is very low and they've dropped them from their entry conditions. So that's good news. Basically, New South Wales and Tasmania also have restrictions entering into their state. I've popped the website up there, and if you need any of these links, I can send them on to Maddy to send on to anyone if they need them. So you will have to meet entry requirements moving into there. Ultimately, we've been preparing industry notices that have gone out to industries, and we've been working with NDIF, AUSVEG and Hydroponic Farmers Federation to try to help inform and keep industries up to date. And if you need any more information or if you need to consign stuff anywhere, this is our group email, which is the marketaccess.agriculture.vic.gov.au, and we can help you with any information you need to potentially move product. Thanks, Maddy.
So I'm Cliff Kinoti, as I said, from Agriculture Victoria, and today I'll be giving an update on important vegetable virus diseases in Victoria. And this is through a project that's Hot Innovation funded, that we've been doing a survey for diseases in Victoria farms. And so far through this project, we've visited 52 farms, that's from since 2018. And sampled more than 500 vegetables and weed samples for virus testing. And in terms of viruses detected across different vegetables, we found a lot of proportion of alfalfa mosaic virus, cucumber mosaic virus, and tomato yellow leaf spotted wilt virus. And also, these are the same viruses we found in a lot of the weeds that we tested. Almost 70% of the weeds we collected, more than 100 had viruses infection. So in this talk, I'm going to cover these 3 viruses. And I'm also going to talk about tomato yellow leaf curl, which Lavinia has touched on. And I'm going to talk more about the biology of these 3 viruses and 4 viruses. And also towards the end, I'll talk about the management strategies. So we're going to start with alfalfa mosaic virus, which is widely distributed across Australia. And here's an electron microscope image of alfalfa mosaic virus, and it has a shape, particles shaped like rod-like. And alfalfa mosaic virus has a broad host range of more than 150 plant species, which include vegetables such as beans, capsicum, lettuce. And also we find it a lot in weeds. You can see the images below. Those are weeds with alfalfa mosaic symptoms. In terms of spread, aphids are what spread alfalfa mosaic viruses, and there are many species of aphids that can spread AMV, and such as the green peach aphid, the melon aphid, and also potato aphids, amongst others. And transmission of alfalfa mosaic virus is a non-persistent manner where the virus is picked by the aphid after short feeding periods. That's probing on infected plants. And then the aphids can be infected for an hour before it loses the viruses. And this is why when you see an outbreak of alfalfa mosaic virus in the field, it occurs as patches in the infected crops in a block. AMV is not seed transmitted in veggies, but there are limited reports of seed transmission in some of the weeds. In terms of disease symptoms, AMV symptoms differ depending on the age of the plant and also the type of the plant. But generally, what you see from alfalfa mosaic virus is characterized by this yellowish or whitish mosaic symptoms on leaves, as you can see here on the lettuce and parsley and some broccoli. And these blotches are the characteristic symptoms of alfalfa mosaic viruses in most crops. In some plants, alfalfa mosaic is associated with severe stunting, as you can see the pea plant on the right-hand side. And in such plants, you see shortening of internodes, which is one of the characteristics of alfalfa mosaic viruses in legumes. So the next important viruses that we found in Victoria is cucumber mosaic virus, which is widely distributed across Australia. And here's an electron microscope image you can see on the top right of cucumber mosaic virus, and it has some round particles. CMV has a large host range, more than AMV, more than 1,000 plant species, and it infects veggies such as cucurbits, lettuce, legumes, and tomatoes, and also is widely found in weeds in Victoria and across Australia. In terms of its spread, it's also transmitted in a non-persistent manner similar to AMV by short feeding periods. And CMV is not transmitted in seed in veggies similar to AMV. In terms of disease symptoms, they vary also in CMV depending on the age of the plant and also the type of plant. But generally, CMV is characterized by mosaic and mottling symptoms on leaves. And in celery, spinach, and tomato, we see symptoms that are characterized by leaf malformation and also shoestring symptoms, where the leaves become tiny and elongated. In lettuce plants, we see twisting of leaves and midleaf browning. And some of the lettuce varieties, you find that they lean to one side, which is a characteristic symptoms of CMV. In most crops, we also see severe stunting, especially when infection occurs when a plant is very young. And in terms of fruit symptoms in capsicum, cucumber, and tomato, we see small, deformed, and ring spot symptoms on fruits, which sometimes can be confused with tomato spotted wilt, which is another virus that cause as almost similar symptoms. So the next virus I'm going to cover is tomato spotted wilt, which belongs to the genus Tospoviruses. And it was first reported in tomatoes in Australia in 1915. That's around Werribee region. And since then, it's found across the world. And it's characterized by these round particles when you look at it under the electron microscope. And tomato spotted wilt is one virus that has one of the largest host range. And it infects commercial hosts such as beans, capsicum, and various ornamentals and weeds as well. In terms of transmission, tomato spotted wilt is transmitted by several species of thrips, but the western flower thrips is the most efficient in transmitting tomato spotted wilt. The life cycle from an egg to an adult takes around 20 to 30 days, but this is wholly dependent on the temperature. If there is higher temperature, the life cycle can be shortened to 10 days or even 5 days. So during the hotter months, we find more and more thrips in the farms. In terms of its life cycle, the thrips acquire the virus through feeding of infected plants at the pupa stage. And once the thrips acquire the virus, they can transmit the virus through its lifetime. And that's why thrips are very efficient in transmitting this virus, and they can end up wiping whole blocks of crop, as you can see in this image of lettuce infected with tomato spotted wilt. In terms of disease symptoms, they vary as well according to the host and also the variety. But generally, we see yellowing, distortion, and necrotic symptoms, which is one characteristic symptoms of tomato spotted wilt in most crops. And early infection, especially in lettuce, you see severe stunting, and there's no heart formation. In plants, in crops like tomato, we see purpling or bronze coloration in leaves, and there's also fruit distortion and ring symptoms in tomatoes and capsicum as well. And early infections, most of the time, leads to severe wilting and plant death eventually. And that's why tomato spotted wilt is very important for veggie crops, and it's a major issue, especially in Victoria, where we've been seeing massive outbreaks through our survey. So the last virus I'm going to talk about is tomato yellow leaf curl virus, which Lavinia has touched on. And unlike the other three viruses that I've talked about, which are RNA virus, tomato yellow leaf curl virus is a DNA virus. And it has a limited host range, about 30 plant species, but it's more damaging because of the yield loss associated with this virus. It infects veggies such as capsicum, chilies, legumes, tomatoes, and also some of the weeds which act as an alternative host for the virus. It's not seed-borne, and it's mainly transmitted by the silver whiteleaf, where the whiteleaf acquire the virus by feeding and transmits the virus all through its life. In terms of disease symptoms, tomato yellow leaf curl symptoms appear 10 to 21 days after infection. And that's why it's more damaging, because infection can go unnoticed until when it's too late. But general symptoms are leaf yellowing and upcurling and standing of plants. And there's no flower formation or fruit formation in plants that are infected when they're young. And we see also enlarged bushy-like leaf in legumes, which is a similar symptoms associated with tomato big bud disease. We also see yellowing mosaic in beans, and in capsicums or cherry plants, they might be symptomless, but they will not produce any flowers or fruits. So, and finally, I'm going to talk about disease management of these viruses. And one thing that we should know that plant viruses, there's no cure and infection is for life. So therefore, it is important to use measures to prevent virus infection and to prevent its spread to other crops. So one of the most important things in terms of virus disease management is removing the source of the virus and the vector in the picture. And this can be through weed control, which an old study, I think 1963, found that leads to a reduction of disease incidence to less than 10% compared to where there is no weed control. It's also important to rapidly remove infected or leftover plants to reduce virus transmission to other healthy crops. And also ensure that the use of virus-free, healthy planting seedlings when you're establishing new crops. There's also other integrated approach for managing viruses diseases, such as vector monitoring using the yellow sticky traps, crop rotation, and general crop or farm hygiene, and also use of biological controls or chemistry to control and put pressure of vectors down. And also, lastly, it is important to identify the pathogen-causing diseases. This is through diagnostic that you can, so that you can employ the best management practices. And in this project, through Agriculture Victoria, we're offering free vegetable diagnostic for viruses, bacterial and fungal diseases, which growers can take an advantage for. So I'd like to acknowledge all these people from Agriculture Victoria, AUSVEG as well, and Horticulture Innovation Australia who are supporting this project. Thank you.
Good afternoon, everyone. My name is Ramez Aldaoud. I'm a plant pathologist with Agriculture Victoria based at Bandura. And I am one of the diagnosticians here today. I'm going to go over a diagnostic result on those samples collected from Werribee, if you remember the workshop in April. And also there were samples submitted afterward. Basically, I'll be focusing on fungal and bacterial pathogen. So that really a footage of that Werribee workshop, previous one, where we did a demo of how to collect a sample and some sample collected and taken with me to Crop Health Service in Bandura. At Crop Health Service, actually, all the samples are databased by Con and Peri and their team, very much to do with the detail of the client and what tests need to be done. And there is a submission sheet coming with the sample usually, and they generate what you call it a worksheet that go to us here, the diagnostic teams. I'm one of them here. So whereby at this stage, sample being investigated, inspected, and test being decided on and allocated. The range of tests that we do there, and it could be one or a combination of any of these, depend on the situation. In this case, most relevant one is fungal culturing for these samples. So the test we do actually mostly, as I said, fungal culturing, bacterial culturing, that's the most relevant one. And we can take it all the way to the molecular level, especially with bacterial one, because species identification and beyond has to be done this way. And Alice is a key person there, plus Cliff as well. This is the results from some of the sample. The symptom is really very much galling of the root system, which is very typical clubroot, and you see it both on adult plant and even young seedling. This is rocket one. Even symptomless root can show this, and this is an example of actual diagnostic of the clubroot fungus affecting occupying the cell infected, and that really masses of resting spore occupying the cell which are affected. This is very easy test. Direct microscopy will do the job. This is another sample whereby we found Fusarium and Microdochium, and just affecting the root system. In this case, cauliflower. Here we have Pythium and Fusarium both found affecting the root system as well. Both of them are soil-borne, and you could see the damage and severity of the damage it could cause to the plant, especially actually when plant are seedling or early stage growth. This is another parsley sample rather, where we got a combination of Pythium, soil-borne pathogen as well, and Mycocentrospora acerina. This fungus, we don't see it that often, and it very much more in coriander and related to plant. And in this case, what we have here is parsley rather. We don't see it in brassica. So far, we haven't seen it so far. Mixed infection could happen as well. This one of the sample when we got Sclerotinia on the same sample, and I'm talking about Broccolini leaves here at the base, and this is an enlargement here, if you can follow me. And also we got Pseudomonas same time from the same sample affecting the foliage here. This image here, this is really from a American website to show you the typical symptom, what we call timber rot disease that caused by sclerotiorum, sclerotiorum. That's the name of the disease because it causes kind of dryness of the root and it becomes more temporary. This is where we got Formalingam, and that's common in brassica and canola, one of them. Plus, in this case, we have the Broccolini. In this case, we got really the sample have this leaf lesion, leaf spot. Initially, the client was thinking of bacteria rather, but no, that what we ended up getting is Formalingam. Formalingam typically cause what we call blackleg, and this image to the left here is also from a website showing you the typical symptom it can cause on the stem and lower part of the plant. This is a lettuce sample. This is an amazing, interesting sample now where we got both Microdochium, which is the anthracnose fungus, and also now we're investigating, still under investigation, a bacteria coming out of it. It's most likely to be Xanthomonas hortorum, but still this to be confirmed. This seedling here of broccoli, we got 3 pathogens here. The key one was, in this case, Xanthomonas campestris, the black rot one, and that different from what we call blackleg. Blackleg is fungal pathogen, black rot is more bacterial pathogen here. We also found Pseudomonas fluorescens and also botrytis in this sample. I believe both of them are secondary, compare or relative to the black rot pathogen, which is Xanthomonas campestris in this case. We're finding, this is a rocket sample, we're finding Pseudomonas viridiflava and other bacteria. This unusual bacteria, somehow Bactobacterium carotivorum, is more the soft rot one that causes potato problem, soft rotting. But this is a species, at the species level, to have it from lettuce, we don't know what the pathovirus there, we couldn't identify. And this symptom is a bit, a bit unusual, and we're not really sure whether really the bacteria causing it per se in this case. Lettuce samples here, we got Xanthomonas hortorum to the left, plus Pseudomonas viridiflava to the left. Pseudomonas viridiflava also we found it in lettuce showing general rot symptom, and also we got Xanthomonas hortorum vitian, the pathovirus, causing unusual symptom, really, rot symptom like this. This is atypical for Xanthomonas in general. This is a coriander sample to the left. We got Pseudomonas viridiflava from the leaves, and also parsnip, where we got also the same pathogen from rather the root, rather the taproot. It's not always easy for diagnosticians. Sometimes we get nothing out of it. You do all the tests, and in the end, you get no really key pathogen. In this case, you get this leaf damages here and rot on already adult plant. To the right, we got seedling as well. And here we have adult plant with some distortion. We've done all the possible tests we could do. Nothing we could come out with. So situation like this for us very challenging, because we don't have anything to say to the grower except they have to think about other non-disease factor, environmental and what have you. Reporting wise, as I said, we've spoken about that before. It really, confidentiality is the bottom line and really the benchmark for us, whether it's informal or formal. And usually suspect exotic, they go within 24 hours. This is our obligation to the Biosecurity Victoria, the Chief Health Officer, plant health officer there, and of course confidentiality meant to be maintained and kept. To summarize, so basically with fungi we found those the clubroot fungus, Fusarium, Microdochium, they all soil-borne, Pseudomonas as well, Mycocentrospora eserina, and they're all soil-borne. Formalingam can be both soil-borne and also airborne. Sclerotinia is mostly soil-borne and depend whether it can develop another stage. We call it ascospores that can go as well as foliar. Botrytis we found it only in one incidence in this case, which is, I believe, it's going to be secondary rather. We found it only on seedling, the cauliflower seedling early on. Bacteria was at the conclusion of those findings that with the sample we received, we diagnosed so far. We get Bacterium, Sclerotium, Pseudomonas fluorescens. Syringae is a key one. Werribee flava, we've been finding more of it. Xanthomonas, the black rat one, is very key in brassicae. And Xanthomonas hortorum vitian is more actually in lettuce, this one. So that's all what I have for you, and I acknowledge that all the team in Crop Health Service and also AUSVEG for their, of course, supporting this work. And thank you.
Vegetable leaf miner, serpentine leaf miner, and American serpentine leaf miner are 3 exotic Luriomyza flies from the Agromyzidae family and are all well-known pests overseas. Vegetable leaf miner, Luriomyza sativii, was first detected in the Torres Strait Islands in 2008 and first detected on mainland Australia in Seychelles at the tip of Queensland in 2015. It is currently under quarantine in the region and hasn't spread further. Serpentine leaf miner, Luriomyza wiedebrensis, was first detected in Greater Sydney in October 2020, followed by the Fassifern Valley in Queensland in November 2020. It has since spread to other regions across New South Wales. Most recently, American serpentine leaf miner, Luriomyza trifolii, was detected in the Torres Strait Islands, Kununurra and Katherine in July-August 2021. None of these species are currently present in Victoria, and we want to keep it that way. However, all 3 species are no longer quarantinable pests, meaning businesses will not be shut down due to detections on farm, but they should still be reported to the Exotic Plant Pest Hotline. Here is an image of a fly against a 5-cent piece, showing how small these flies really are. Leaf miners can have a dramatic impact on plant health. Eggs are laid just beneath the leaf or stem surface and hatch about 3 days later. Once hatched, larvae tunnel inside the leaf, creating silvery spiraling tracks. Here is a video of a leaf miner larva tunneling through a leaf. It is the most disruptive stage of the life cycle. There are 3 larval stages that occur within the leaf, and once the 3rd instar larvae has developed and matured, it will exit the leaf and pupate in the soil. Here you can see leaf miner pupae and an adult leaf miner emerging from a pupa. Adults typically emerge from pupae after 7 to 14 days. The adult stage is the final stage of the leaf miner life cycle. This image shows a yellow sticky trap with thousands of adult serpentine leaf miner, and this image was taken in Sydney 2020. So, what's the problem with leaf miners? Well, they damage photosynthetic tissue, restricting plant growth, and they can also cause crop failure or kill the plant entirely. The left-hand image here shows leaf mining damage to a seedling. The other 2 images show other damage known as stippling. Stippling refers to the holes caused by adult flies when feeding and laying eggs. Exotic leaf miners are polyphagous, meaning they will eat most vegetables and ornamental flowering plants. They are also known to harbour in weeds. So what does this mean? Well, Australian horticulture is currently valued at $14.4 billion. When we find a new pest in Australia, the economic damage to production industries can be substantial. To put this into perspective, serpentine leaf miner caused a 70% yield loss in potatoes in Indonesia in 1995, and American serpentine leaf miner cost Californian greenhouse ornamental growers $21 million per year in the 1980s.
But what is causing such high losses? To answer this question, I'll start with these two photos of bean plants in South America. One of these was regularly sprayed with insecticide and the other was not. And can you guess which one is which? Counterintuitively, it is the infested plant on the right which received the treatment. This happens because in a natural system, leaf miner populations are held in check by their natural predators, parasitoid wasps. These wasps are extremely sensitive to chemicals, and inappropriate insecticide exposure will decimate populations. While these insecticides may reduce leaf miner populations temporarily, they will recover quicker than the parasitoid wasps, and populations will grow uninhibited by predators. This is an example of a secondary pest, one where the damage isn't problematic until something disrupts the system, in this case, a chemical application. So what's happening in Australia? In Australia, serpentine leaf miner was detected in Greater Sydney in October 2020. Some of the badly affected crops included beans, cucumber, chilli seedlings, parsley, and lettuce. It was determined that this pest was unable to be eradicated from Australia, and there was a move to management. The pest then spread across New South Wales and was detected in the Fassifern Valley in Queensland in November 2020. Since then, the distribution has not largely changed, but it is likely to spread through human means as they are not strong flyers. In July 2021, American serpentine leaf miner Liromyza trifolii was detected across 3 locations in Western Australia, Queensland and Northern Territory. It has been declared not technically feasible to eradicate, but it is under containment in the region. In terms of management, there are still many unknowns about how leaf miners will behave, and we need at least 2 to 3 seasons to better understand their behaviour. From international experience, we know that integrated pest management is critical for appropriate control of these pests. Biological control will be the major component of the integrated approach. Many of the specimens collected during the response to last year's serpentine leaf miner detection had evidence of parasitism by endemic parasitoid wasps. In this video, we can see a parasitoid wasp attacking a leaf miner larva. The parasitoid wasp is the dark image and the leaf miner larva is the yellow on the left. Industry partners have put together preparedness plans and management plans on these pests to help support growers in preparing and managing these pests. In summary, exotic leaf miners are found in Australia, but they are not found in Victoria. This means that there are no longer quarantine restrictions associated with these pests, but we encourage industry to report them to the Exotic Plant Pest Hotline on 1800 084 881 so we can detect them and manage them early.
Well, thank you to everybody who stayed online for that video. If you have any questions about exotic leaf miners, please get in contact with me at AUSVEG. The best contact email is science@ausveg.com.au. Once again, thank you to all of our speakers today, and thank you very much to our participants. We hope you found this webinar valuable and look forward to seeing you at our next Peri Urban workshop. Thank you.